These 4 papers from the June 2026 issue of Biological Psychiatry are best read together. None of them solves depression, but each opens a different window onto the same hard problem. Depression is not one disease. It's biologically heterogeneous, and that heterogeneity is exactly why we've struggled to find biomarkers and targeted treatments. Here's the framing that ties all 4 together. Depression affects more than 300 million people, and the WHO projects it'll be the leading contributor to global disease burden by 2030. Yet, after 40 years and 15,000 neuroimaging studies, no single brain region, when lesioned, reliably causes depression. The DSM lets you reach a diagnosis through 227 different symptom combinations. 2 patients with the same label can share 0 symptoms. So the field has shifted from asking, where is depression, to asking, what are the systems, networks, genes, molecules, cells, whose disruption produces these many depressions. Our 4 papers each take one of those systems. The first paper, by Baller and colleagues, is a review with a clever proposal. If no single lesion location causes depression, maybe lesions in different places that hit the same network do. That's the logic of lesion network mapping. Their move is to use multiple sclerosis as a natural model. MS produces white matter lesions. Up to half of patients develop depression, and critically, these patients already get annual MRIs through routine care. So instead of expensive research scans, you have a massive insurance-funded imaging archive. They show MS lesions preferentially fall within a white matter depression network, and they lay out a roadmap from association toward causality, longitudinal designs that track whether a new lesion brings on new depressive symptoms. The second paper, Mitchell and colleagues, zooms out to the genome. The latest consortium, GWS, found nearly 700 genetic associations with depression, up from just 2 a decade ago. Why does it take 685,000 cases to find signal? Because depression is extraordinarily polygenic, and the authors argue that polygenicity is partly a symptom of our messy phenotype. Their review organizes genetic heterogeneity across 5 domains: symptom profile, environment, age of onset, sex, and treatment response. The takeaway for us: a typical depression looks metabolically distinct. Early onset looks more heritable and neurodevelopmental, and lumping everyone into one MDD case definition dilutes the biology. Cleaner phenotypes, not just bigger samples, are the path forward. The third paper, Nisbet and colleagues, asks the obvious next question. Those GWAS hits are just genomic coordinates. What do they actually do? They used a method called OPERA to integrate 5 molecular layers at once: gene expression, methylation, splicing, chromatin, and protein abundance, across blood and brain. Most prior studies looked at 1 layer at a time. Integrating them gives stronger causal evidence. They flagged 23 high-confidence genes supported by 3 or more molecular layers, including known players like CDH13 and a novel hit, H6PD. And those genes were enriched for druggable targets, which is really the prize here, turning statistical signal into something a chemist could act on. The fourth paper, Iredi and colleagues, gets even closer to the cell. It's the largest blood transcriptome mega-analysis in depression. About 6,000 people across 5 data sets, harmonized, and, crucially, corrected for cell composition, which most earlier work skipped. Their key insight: once you account for which cells are in the sample, the signal points to lymphoid cells, T cells, and natural killer cells, and to a drop in replication-dependent histones, with a female-specific oxidative phosphorylation effect. This converges beautifully with the genetic evidence that depression risk variants cluster in active sites in T cells. 2 independent windows, genes and blood RNA, pointing at the same immune story. So step back. 4 papers, 4 scales, circuits, genes, molecules, cells, and 3 threads run through all of them. First, heterogeneity is the central obstacle, not a nuisance to average away. Second, convergence is the validator. When network mapping, genetics, multi-omics, and transcriptomics independently implicate immune and metabolic biology, we should believe it. And third, every group reached for scale through existing data, clinical MRIs, biobanks, public X, QTL, and expression datasets, because that's how you get the power to resolve subtypes. The clinical promise is real: biologically defined subtypes, and the druggable targets to match them.